So let us look at what we term as enzyme inhibition. We want to look at the different chemicals that can actually alter enzymatic action. That is, they make enzymes not to basically work. So we want to look at how these inhibitors work and what are the implications as far as changing the concentration of the enzyme or changing the concentration of the substrate is concerned in relation to the action of inhibitors. So we start by looking at this scenario. So in this scenario, what we are basically having is that you have a pharmaceutical company that is developing a new drug to manage a problem. This is a rare metabolic disorder that is caused by an excess production of a compound. And that compound is zeta-7. Okay, so we shall call that compound zeta-7. So this disorder, this metabolic disorder, results from overactivity of an enzyme called Z7 synthase. So this enzyme catalyzes the final step of formation of zeta-7. Okay, so we are having in the body, we have a lot of zeta-7, but we have an enzyme that catalyzes the final reaction for forming that zeta-7. Now, to solve the problem, if the scientist can actually inhibit the action of this enzyme, it means that we shall not have this zeta-7. Okay, so what the scientists are actually doing is that they are testing two inhibitors. So one inhibitor, that is inhibitor A, is a molecule that structurally resembles Z7 synthase. Okay, and then the second one, which is inhibitor B, is a large molecule that binds elsewhere on the enzyme and alters the active site. Okay, it does not bind to the active site, but binds somewhere else, but the result is that it changes the active site. Okay, so clinical trials, that is the different, I'm calling them clinical trials, but basically the laboratory trials have shown that inhibitor A is effective in small doses, but becomes less effective when you increase the concentration of the substrate. Okay, but B actually works smoothly or steadily across the different substrate concentration. Okay, though its effects are harder to reverse. So, like again, we said earlier on, in real life, in science especially, we have to make choices. And these choices are actually sometimes very hard to make because you not always have everything on the silver plate. So you have two inhibitors, and you have to choose which one works better. They have different qualities or characteristics. So let us look at the questions. The first question is, can you explain the types of enzyme inhibition exhibited by inhibitor A and inhibitor B? Can you support your answer with diagrams and relevant molecular level details? Okay, can you provide that molecular level detail about enzymatic inhibition illustrated by inhibitors A and B? Secondly, can you compare the effects of competitive and non-competitive inhibition on enzyme kinetics, issues to do with the rates of enzymatic activity in relation to whether competitive or non-competitive inhibition? And then lastly, can you suggest which inhibitor would be appropriate for long-term therapeutic use? Well, using this to treat people who have these illnesses over an extended period of time, especially this chronic metabolic disorder. Can you justify your choice using biological and medical reasoning? Okay, which of the two inhibitors can you use to treat this long-term condition? Is it A, is it B, and why? Okay, and then the last point, can you discuss the potential risks associated with prolonged enzyme inhibition in a human body and suggest how this might be monitored or minimized in drug development? Okay, so this is an excellent question. What this question basically hints on are the issues to do with enzymes and how our knowledge of enzyme activity can be actually applied in addressing some of the medical problems that can happen in humans. We already saw that actually enzymatic activity can be applied in managing wastes. And now we are looking at how enzymatic activity can be used in managing certain conditions. Okay, of course, in our bodies, we have metabolic processes that happen. And these processes give us products. Some of these products that come out of the metabolic process are actually dangerous to the human body if they are not reduced to other components, or if they're not broken down to other components that are quite safe. So that means that if people don't have the enzymes that can break these compounds to relatively safe molecules, then they are going to have problems, because these toxic molecules can actually build up in the body, and then you have problems. And for example, just a simple illustration that, for example, if you break down proteins, you get ammonia. And if you don't have the enzymes to convert that ammonia into urea, then you will have problems, right? This is the same thing that we are talking about here when you have a metabolic disorder because of lack of particular enzymes. Or when you have an enzyme that is working very hard and then producing a molecule that will actually turn out to be toxic to the body, then that means that you need to deal with that enzyme and make sure that it doesn't work, such that you reduce the product that it is producing that is actually toxic. So that is the logical reasoning behind this question. Okay, so let's look at the presentations and then later on come back to the questions on this slide. So when we talk about enzymatic inhibition, we are talking about molecules or substances that can actually reduce the activity of enzymes. And these molecules can be actually organic or inorganic, and then causing this disruption in enzymatic activity. So some of them can bind to active sites of the enzyme, others do not necessarily need to bind to the active sites of the enzyme. And therefore, you have inhibitors that are termed as competitive inhibitors if they are looking for the active site of the enzyme and then causing it to slow down. And then we have those that are non-competitors. For them, they don't look for the active site. They bind on another site on the enzyme and then reduce its action. Okay, so those are the two categories. And then you have what we call reversible and irreversible competitors, inhibitors. With this, we are talking about those that can permanently alter the enzyme's activity. That means that if that inhibition happens, it's permanent, it's not temporary inhibition. Okay, so that's where you have reversible versus irreversible, and usually that comes in with the strength of attachment between the inhibitor and the enzyme. If then that attachment is a strong one, then that means that it's going to be permanent or irreversible inhibition. If it is temporary, then it is going to be reversible. Okay, so temporary is what I've just explained. Irreversible, strong bonds between the enzyme and the inhibitor, and therefore you can't have separation of the enzyme with the inhibitor, and therefore you're going to have permanent inhibition there, which is basically irreversible. Because if that separation happens, they damage the enzyme and the enzyme will no longer be able to work. Okay, so competitive inhibition, the inhibitor has to resemble the natural substrate and in terms of structure, so that it can fit well into the active site. And then you have some examples there. So competitive inhibition resembles the usual substrate such that it can occupy the active site. Okay, so the example that we have is what is illustrated on the screen. Okay, so you can see that the substrate, if the substrate is not there, you can have the inhibitor occupying the active site, and then the enzyme will no longer, the enzyme substrate complex will not be formed because you have an inhibitor taking the place of the natural substrate. Okay, so when you have the inhibitor occupying, you cannot have the substrate occupying the active site, and then you have reduced enzymatic activity. The key thing here is that for competitive inhibition to be effective, you need to have a higher concentration of the inhibitors. Okay, such that the inhibitors can outcompete the substrate and then form and have that binding. But if you have less of the competitive inhibitors, then that is when you're going to have a problem with this inhibition, because it will not be effective, because the substrates will be more compared to the competitive inhibitors. Okay. Okay, so for non-competitive inhibitors, what basically happens is that they do not bind on the active sites, they abide on another part of the enzyme, and then this alters the active site such that the substrate can no longer fit into the active site. Okay, so for it, the unique thing with it is that it is not dependent on the concentration of the substrate. So that means that you don't need to have more of these non-competitive inhibitors to have the effect that you want. Because as the substrates are competing for the active site, this one competes for another site, and then altering the whole thing altogether. So that is what happens. And cyanide is an example of this. Okay, cyanide is a respiratory poison or toxin, or basically inhibitor. What it does is that it attaches on another side of the enzymes that we call the cytochrome oxidases, and then you don't have the usual substrates could now binding to the active sites of these enzymes. Okay, so they do not depend on the substrate concentration.